Every living thing, from the smallest little bacterium to a massive blue whale, is built from a set of instructions, a master blueprint. Today, we're going to unpack that blueprint and look at the absolutely incredible architecture of DNA.
So if you could just unroll the complete instruction manual for a person, what would you even see? Well, it's not paper and ink. It's a molecule, and its structure is honestly one of the most beautiful and important discoveries in the history of science.
All right, so to really get our heads around this amazing structure, we have to start with the basics, right? Before we can build the whole thing, we need to look at the raw materials. What are the actual bricks and mortar of life?
Okay, so at its heart, DNA is a polynucleotide. Now, I know that sounds super scientific, but all it really means is that it's a very, very long chain. Seriously, just picture a necklace made of millions and millions of tiny little beads. DNA is a polymer, just a massive molecule built from these repeating smaller units, which we call nucleotides. Whoa, okay, I know this diagram looks pretty complicated, but stick with me. It's simpler than it looks. Each one of those links in the DNA chain, each bead on our necklace, is made of just 3 parts. You've got a phosphate group, a sugar called deoxyribose, and then a nitrogenous base. That's it. That's the core pattern: phosphate, sugar, base. Just repeated millions of times over to make the chain.
Now you can imagine that drawing out that full chemical structure every single time would be a total nightmare. So scientists came up with a shorthand. That whole complex chain of phosphates and sugars just becomes a simple line. That's the backbone. And the really important part, the base, is just shown by its first letter, A, G, C, or T. This simple little stick figure is what we're going to use to build our masterpiece.
For the longest time, scientists knew all these pieces were there, but they had no idea how they actually fit together. It was a huge puzzle. That is, until the 1940s, when a biochemist named Erwin Chargaff found a critical piece of that puzzle. So Chargaff did this incredibly detailed work, analyzing DNA from a whole range of different species, and he found this bizarrely consistent pattern. It didn't matter if it was from a person or a tiny bacterium. The amount of adenine, or A, was always the same as the amount of thymine, T, and the amount of guanine, G, always perfectly matched the amount of cytosine, C.
This was just too perfect to be a coincidence. It was a massive clue, and it pointed to one obvious conclusion. These bases must physically pair up with each other. Adenine has to connect with thymine, and guanine has to connect with cytosine. This simple little pairing rule was the key that finally unlocked the entire structure of DNA.
So, how does this one rule build the entire final structure? Well, armed with Chargaff's discovery, 2 scientists, James Watson and Francis Crick, were finally able to put all the pieces together. And in 1953, they showed the world their groundbreaking model for the architecture of DNA.
And here it is, the famous double helix. It's a right-handed helix, which means it twists to the right, kind of like a corkscrew. The easiest way to think about it is like a twisted ladder. This elegant structure became a true icon of modern biology, and for good reason.
So let's look a little closer at this architecture. You've got these 2 long strands twisted around each other. The sugar-phosphate chains we talked about earlier, those form the sides of the ladder. And the bases, the A, T, C, and G, they form the rungs, and they're tucked safely inside. And what's really crucial here is that the 2 strands are complementary, not identical, all thanks to Chargaff's rule. The strands are also what we call anti-parallel. This is a really key feature. The best way to picture this is to think of a 2-lane highway. You've got cars in one lane going north and cars in the other lane going south. The 2 DNA strands are just like that. They run in opposite directions.
So how do those rungs of the ladder actually stick together? Well, if you look at these diagrams, you can see the bases are connected by what are called hydrogen bonds. You can think of them as tiny little magnets holding A to T and G to C. And because of the specific shape of these bases, this is the only pairing that fits perfectly inside that double helix.
But here's a really cool detail. Not all of these connections are equally strong. An AT pair is held together by 2 of these hydrogen bonds. The GC pair, though, has 3. That makes the GC bond about 50% stronger. It's a small difference, but it has huge implications for how our cells read and copy our genetic code. Now you might be thinking, okay, double helix, story's over. But nope, not even close. It turns out that under different conditions inside the cell, this amazing molecule can twist and fold into a bunch of other fascinating shapes.
The classic Watson and Crick model we've been talking about is called bDNA. It's the most common form in our cells, but there's also aDNA, which is a bit shorter and wider. And then you've got the real rebel of the group, zDNA, which is thinner and actually twists to the left in this funky zigzag pattern.
And if you thought 2 strands was the limit, get this. DNA isn't always a double helix. Under certain conditions, a 3rd strand of DNA can actually slide right into one of the grooves of the helix, forming a totally stable, triple-stranded structure. And believe it or not, it can get even more complex. In parts of our DNA that are packed with the base guanine, 4 strands can actually come together and form this stable, box-like structure called a G-quadruplex. It's almost like a tiny molecular knot. So why does any of this matter? Why do we care about all these different shapes? Well, because they aren't just scientific curiosities. These G-quadruplexes, for example, are often found at the very ends of our chromosomes, in regions that are super important for cell aging and for cancer. Scientists are now designing drugs that specifically target these weird structures, opening up completely new ways to fight diseases. The blueprint of life, it turns out, has more than a few hidden rooms.